Ms. Ayla Zhang, and today I will be presenting my paper evaluating cancer drug targeting pathways with large language models. And this study presents a novel framework combining bioinformatics tools and AI-driven evaluations to streamline target identification using pancreatic adenocarcinoma as a case study. So pancreatic adenocarcinoma, or PAAD for short, is a highly aggressive solid tumor that accounts for over 90% of all pancreatic cancers. And PAAD is one of the most challenging malignancies to treat with a 5-year survival rate of less than 10%. And it currently ranks third in cancer-related deaths in the United States, with projections indicating it will become the second deadliest cancer by 2030. And at the time of diagnosis, over 80% of patients have experienced metastasis and spread of disease, evidencing PAAD's poor prognosis and resulting in advanced tumors that are ineligible for surgery. So some traditional treatments for PAD, such as chemotherapy and immunotherapy, have failed to produce meaningful results measured by patient survival because of the immunosuppressive and very desmoplastic tumor environment in PAD. So, for example, some approved chemotherapies such as gemcitabine or FOLFIRINOX only improve survival by 2 to 4 months and are usually accompanied by severe toxicity and adverse effects. And then some current targeted approaches to PAD drug development have been researched extensively with the availability of cancer genomic sequencing data. So, for example, KRAS, which is mutated in nearly 90% of PAD patient samples, has become a common target for investigational drugs. However, leading drugs involved in clinical trials for PAD treatments, such as sotorasib, have also shown minimal survival improvements around 3 months and also commonly include adverse effects. So therefore, recent research regarding PAD drug targets has shifted towards genomic tumor drivers that are predominantly involved in PAD signaling pathways. So this paper describes the bioinformatics process of ranking potential PAAD targets with a systematic, unbiased approach by evaluating their relation to PAAD. And to achieve this, we implemented an innovative analytical framework. So the approach that we developed has the following innovative characteristics. First, we performed a comprehensive pathway analysis to assess a drug target's relevance to PAAD progression and suitability as a novel target for PAAD treatment. Second, we leveraged ChatGPT 4.0 in our evaluation tasks to improve efficiency and demonstrate the utility of generative AI in de novo drug discovery. Finally, we streamlined the drug discovery process by integrating this ChatGPT evaluation with the bioinformatic pathway analyses to rank the potential drug targets. So our methodology was structured into 5 steps to systematically evaluate and prioritize candidate drug targets for pancreatic cancer targeted therapy. And the first step in our process was to establish a list of candidate drug targets. We compiled an initial list of 252 genes that are members of pancreatic cancer-related pathways, annotated lists, and gene signatures, also known as PAGs, identified using Pager, which is a pathway analysis tool. So this image is an example of a gene signature found on Pager related to pancreatic cancer and shows these genes listed as PAG members. To rank the candidate drug targets, we established a comprehensive evaluation process that assessed each candidate in relation to PAAD. So instead of directly assessing the drug targets themselves, we retrieved and evaluated biomolecular pathways that are impacted in PAAD and also related to each candidate. And this strategy was designed to ensure that candidates with limited research literature in the context of PAAD were not overlooked, and this helped us facilitate the discovery of novel drug targets. So to do this, we queried our initial candidate list from the previous slide, into Pager and compiled only the resulting pathway type PAGs with at least 5 genes overlapping with the candidate list. And this resulted in 363 PAAD related pathways. We then utilized ChatGPT 4.0 to develop a comprehensive evaluation framework to assess the significance and influence of a specific biomolecular pathway in pancreatic cancer. Previous research has validated ChatGPT's consistent performance in evaluation tasks compared to human evaluators and also its reliability in medical and biological contexts, which makes it an efficient and accurate tool for our analysis. So we use a numerical scoring system to minimize potential errors such as hallucinations and ambiguities commonly associated with AI. And the ChatGPT 4.0 framework evaluated each pathway across 7 categories to assess its role in PAAD progression and its potential as a drug target. To evaluate each pathway, the pathway name and the scoring system were inputted as prompts for ChatGPT 4, which then would provide the evaluation score. And these prompts were slightly varied between trials to ensure robustness and improve the reliability of the evaluation scores. So each pathway was scored on a scale from 0 to 9 in each category, with 0 indicating no relevance or evidence, and 9 indicating strong, well-supported significance. And these scores were multiplied by specific weights that were assigned to each category based on their relative importance in determining a pathway's therapeutic potential. So the first category was pathway relevance to PAAD, and it was assigned the highest weight of 5 because it directly evaluates the pathway's involvement in PAAD-specific processes, such as tumor initiation, progression, and therapeutic resistance, which are key factors in identifying actionable targets. And this category was evaluated by analyzing the pathway's documented involvement in key biological processes. Next, target druggability was weighted at 5 also to emphasize the practical feasibility of translating pathway findings into therapeutic strategies. And this was assessed by the presence of actionable drug targets within the pathway, as well as the availability of drugs that are targeting these compounds. Resistance mechanisms are crucial for overcoming therapeutic challenges and improving clinical outcomes. So this category was weighted at 4, and it focused on the pathway's involvement in resistance to existing therapies for PAAD. So pathways with strong evidence from clinical or preclinical studies linking them to therapy resistance scored higher, while those with minimal or inconclusive evidence scored lower. Biomarker availability was also weighted at 4. It is important for tailoring treatments and monitoring therapeutic responses, and it assessed whether the pathway was associated with reliable markers for patient stratification or treatment monitoring. And pathways with clinically validated biomarkers would receive higher scores, while those with experimental or unvalidated biomarkers would score lower. Genetic alterations in the pathway were given a weight of 4 due to its role in indicating the fundamental molecular drivers of PAAD and providing a foundation for stratifying and targeting patient subgroups. Genetic alterations was evaluated by examining the frequency and impact of mutations in pathway-associated genes among PAAD patients. And so pathways containing genes with high mutation prevalence, such as KRAS, would receive higher scores, especially if those mutations are linked to tumor progression or poor prognosis. Literature evidence helps contextualize findings, but it is inherently dependent on research focus, which could bias representation. So for that reason, it was weighted at 3. And literature evidence considers the depth and breadth of research supporting the pathway's relevance to PAAD. So pathways with extensive support from clinical trials or high-impact publications would receive higher scores. And finally, safety profile was weighted at 3 because it may be less determinative in early stage target identification than the other metrics. And this was evaluated by looking at the feasibility of targeting the pathway based on known or anticipated side effects. So the total score for each pathway was calculated by summing the weighted scores across all categories, and these totals were used to rank the pathways according to their overall potential as drug targets. Then the scoring process was repeated 20 times for each pathway, and an average score was taken to find the final ranking score for each pathway. To ensure robustness, we evaluated the scoring framework's sensitivity to changes in category weights. So the pathway relevance and druggability weights varied by 10%, but we found that the overall rankings remained consistent for the top 10 pathways, demonstrating stability in the scoring. To provide a reference point for pathway evaluation, we generated a list of random genes not included in the candidate gene list from step 1, and this was generated using ChatGPT 4.0. And this gene, which we called unrelated genes, was subsequently queried into Pager to identify 129 pathways that are unrelated to PAAD, and these were called the unrelated pathways. So then we apply the ChatGPT 4.0 scoring system to each unrelated pathway using the same methodology for the PAD related pathways. So each of the unrelated pathways was evaluated 20 times, and the average score for all trials was calculated to determine the final score for each unrelated pathway. And then we averaged all the unrelated pathways to establish the pathway baseline score, which we use as a benchmark to assess the significance of PAD-related pathways. Each candidate's gene's membership in these previously evaluated PAD-related pathways was recorded, and 2 distinct scores were assigned for each instance of a gene being part of the pathway. The rank prioritization pathway score, or RPP score, and then the rank prioritization gene score, or RPG score. So the RPP score was derived by dividing the pathway ranking score obtained through the ChatGPT 4.0 evaluation by the pathway baseline score. And this calculation standardized the pathway's significance relative to the baseline score. Then the RPG score corresponded to the rank prioritization score of the gene within that specific pathway, which was found from the Pager database. And this score directly reflects the gene's importance or contribution to that specific pathway. Both the RPP and RPG scores were normalized from a scale of 1 to 0 or 0 to 1. And then the final score for each gene pathway combination was calculated using a weighted formula, with the RPP score contributing 20% and the RPG score contributing 80%. To consolidate all instances of a gene across PAD-related pathways, the average final score for all instances was calculated to derive the gene's overall ranking score. And this comprehensive approach ensures a balanced assessment of pathway level and gene-specific contributions, effectively prioritizing the genes that demonstrate the most significance within the most impactful pathways. So Figure 1 highlights the top 20 pathways identified through our pathway evaluation, ranked by their average final scores. And the top 3 pathways, pancreatic cancer, pancreatic adenocarcinoma pathway, and Homo sapiens pancreatic cancer, demonstrate the strong association of these pathways with PAD progression and therapeutic relevance. And these pathways integrate key signaling mechanisms such as PI3K, AKT, and MAPK signaling, which are also independently ranked in the analysis. And the prominence of these pancreatic cancer-specific pathways in the rankings validates the scoring framework that we used and emphasizes the biological relevance of these pathways in PAD progression. Next, Figure 2 visually illustrates the differences in mean scores and variability between the two groups, highlighting the significantly higher score for the PAAD-related pathways. So we conducted a 2-sample independent t-test to compare the mean scores between unrelated pathways and PAAD-related pathways. And the t-test revealed a significant difference between the two treatments, indicating that the PAAD-related pathways had a significantly higher mean compared to the unrelated pathways. So to quantify the magnitude of this difference, we calculated Hedges G, which is accounting for the two groups' unequal sample sizes. And the effect size that we found, which was G equals 1.24, indicates a significant difference, emphasizing the greater relevance of PAAD-related pathways compared to the unrelated pathways, and also validating the accuracy of our scoring framework. Following the scoring and ranking process, we integrated the RPP and RPG scores, which allowed for a comprehensive evaluation of gene significance. So Figure 3 highlights the top 20 gene candidates, representing the genes with the most substantial and most consistent contributions to high-scoring pathways that are critical to PAAD progression and therapeutic resistance. And many of the top-ranked genes all play key roles in cell proliferation and genomic stability, which are processes heavily implicated in PAAD. And also several genes with less explored roles in PAAD, such as SSTR4, KCNH2, and SRC, emerge as high-ranking candidates, which suggest novel avenues for further research. The gene ranking results closely align with our pathway analyses, reinforcing the biological significance of these findings. For example, EGFR, AKT1, and PIK3CA are closely tied to the top-ranking PI3K-AKT signaling pathway, highlighting their role in cell proliferation and survival. And these associations underscore the integration of gene and pathway analyses in identifying biologically relevant targets for PAAD, providing a robust framework for prioritizing therapeutic interventions. And sensitivity analysis confirmed that minor weight changes did not significantly impact these rankings. So, for example, a 10% change in the weight of pathway relevance shifted these rankings by 3.87%, demonstrating the reliability of the scoring framework. So overall, in this study, we developed a novel framework for prioritizing therapeutic targets in pancreatic adenocarcinoma, and we focused on biologically relevant pathways rather than relying solely on genetic mutation prevalence. And a key innovation in this study was our use of ChatGPT 4.0, which systematically evaluates pathways across multiple categories, accelerating the target identification process. And the integration of ChatGPT 4.0 into our evaluation framework underscores the practical utility of AI to systematically identify and prioritize drug targets. So this AI-driven approach allowed for rapid evaluation across predefined metrics, significantly accelerating the target discovery process. And our findings also highlight the importance of targeting genes with overexpression and high network centrality rather than frequently mutated genes. And this approach has potential applications in other cancers where actionable targets are difficult to identify. However, the limitations of AI-driven methodologies must be acknowledged to contextualize their findings. And the accuracy of AI outputs is inherently tied to the quality, diversity, and representativeness of its training data. So while this study utilized curated data sets such as PAGER to reduce the biases, the underrepresentation of specific pathways or populations could still influence the results. AI frameworks also often simplify complex biological interactions. So while the weighted scoring approach we used allowed for a structured evaluation of the pathways, it may not have fully captured the intricate nature of tumor microenvironment factors or the dynamics of therapy-induced resistance mechanisms, etc. Although our predictions should be further validated to using experimental data or established biological knowledge, our method can also be generalized across a variety of diseases to streamline the discovery and validation of therapeutic targets. And also, emerging versions of ChatGPT and other AI tools, such as specialized models for bioinformatics or drug discovery, could enhance the precision and contextual accuracy of the pathway evaluation. And we would like to acknowledge Adrian Gu of Alpha Mind Club for preparing part of the drug candidate dataset. And we would also like to thank Dr. Xiang Ling for proofreading the statistics section for accuracy. Thank you for listening.